それではマックス・クーパー博士をご紹介いたします。Dr. Max Cooper, please just go on to the stage, please.
Thank you very much for your very kind introduction. I'm deeply honored to be sharing this JapanPrize this year with Jacques Miller. And I appreciate the very kind introduction. We first learned indirectly of B cells through their products, antibodies. First with the demonstration of antitoxins that could confer humoral immunity by von Behring and Kitasato. Later we learned that antibodies were gamma globulins when Tiselius and his colleagues showed that you could separate them on the basis of charge, and that plasma cells make antibodies, something that was sealed when immunofluorescence could be used to show definitely that plasma cells make antibodies. Agamma-globulin-anemic boys were then discovered, and they were shown to have no plasma cells, which made sense if plasma cells made antibodies, and they were vulnerable to repeated infections, particularly to bacteria. Antibodies were later shown to have both structurally, to have heavy and light chains, and Burnett gave his clonal selection theory of how diversity evolved in antibodies. At this point, in about 1961, Jacques Miller made his revolutionary discovery of the thymus being important in development of immune function. And as he mentioned earlier, Jim Gowans, who used radioactive materials to trace the circulation of lymphocytes around in blood and back through lymphatics, and using labels of DNA, radioactive labels, he could show that circulating lymphocytes became plasma cells. Now, there was another interesting finding that wasn't noticed very much by immunologists because it was published in Poultry Science, that the removal of the hindgut lymphoid organ bursa of Fabricius could really wreck the production of antibodies. And this is how the immune system developed was generally perceived at that time. It had been shown that you could take the thymus at a time that it was mainly epithelial, grow it in culture, and it would generate lots of lymphocytes, which Dr. Miller had shown were seeded to the periphery, and with the demonstration that that could become, those could become plasma cells, this was the single lineage pathway that was generally accepted. That could explain fairly easily the defects in the boys who were born without the ability to make antibodies, a block in this last stage of differentiation in this single lineage model. But there were other observations that suggested that might be an oversimplistic view of how the immune system developed. A clue that we used were patients with Wiskott-Aldrich syndrome, another defect that was transmitted in an X-linked fashion. And it was characterized by eczema and platelet deficiency and infections. But with time, these patients became very lymphopenic. And had defective cellular immunity. A simple infection with herpes simplex, a fever blister, could result in a lethal spread of the virus infection in these boys. Interestingly, even though they had an abundance, very few lymphocytes and an atrophic thymus, they had lots of plasma cells and they made high levels of immunoglobulins, which was not so easily explained by this single lymphocyte lineage model at the time. So as I mentioned earlier, Bruce Glick and his colleagues had shown in a serendipitous fashion that removal of the bursa at the time of hatching in chicks could stunt their antibody responding capability. So they had both a thymus and a bursa, so this seemed possibly to be a model that one could go back and look to test whether there was one or more lymphocyte lineages. And indeed, Schoenberg and Warner in Australia had shown that if you inhibited development of this bursa with a high dosage of male hormones, testosterone, you could also reduce antibody production. And in some of the chickens so treated, they had thymic poor development as well. And they didn't reject skin grafts as well. These results were hard to interpret in the light of the work that was being done in mammalian species, and also somewhat difficult to reproduce, in part perhaps because we knew by that time that the status of the immune system development at birth, or hatching in this case, varied. And if you had already well developed your peripheral or your lymphocyte population, that you couldn't just by simple thymectomy or bursectomy in this experimental model show strong effects that were interpretable. So it seemed clear that there needed to be some way to go back and restart or set the clock back in immunological development. And so if it were possible, if one could take out the thymus early in embryonic development, and in others, take out the bursa early in embryonic development, you would have a much clearer effect of what these two tissues were doing in immune system development. The only way that we could think of to do this at the time, since we couldn't do surgery early enough during development, was to try to destroy the cells that had already come from or had been influenced by the thymus or bursa at the time of hatching from the egg. And so we tested the levels of radiation that newly hatched chicks could tolerate and gave them the irradiation and either took out the thymus or the bursa or both or neither, allowed them to grow up and recover from the irradiation effects, and then tested their immune functional capabilities. And the answer was clear-cut at that time. In the bursectomized and irradiated chicks, there was normal development of lymphoid cells or numbers, but they had no germinal centers or plasma cells, not any that you could find, and they made absolutely no antibodies. Whereas the thymectomy in irradiated recovered animals looked like thymectomized mice. They had depletion of the lymphocytes elsewhere in the body. They couldn't reject grafts from other chicks normally, outbred chicks, and they had delayed type hypersensitivity impairment, and also their cells couldn't cause an attack, a graft versus host attack on other animals. But they had germinal centers and plasma cells, but nevertheless, they didn't make antibodies well to all antigens. Now, that led to this model of two lineages, one derived from the thymus and the other from the bursa. And we could indeed put back the bursal cells after irradiation and restore the germinal centers and plasma cells and production of antibodies. So this cartoon just tries to illustrate that these were two lineages coming one from the thymus, the other from the bursa, being replaceable by cells from the bursa if you eliminated them, that were intermixed in spleen and other peripheral lymphoid tissues. One was responsible for cell-mediated immunity and the other for humoral immunity. There's some question marks at this time, of course, because the hematopoietic stem cells had been shown by Till and McCulloch to have multi-potential differentiation. They could become different cell types. And also it had been shown in England that some of these offspring could go to the thymus and become lymphocytes. It was later, very soon thereafter, Owen and Moore showed that there were common progenitors that could migrate to either the thymus or the bursa. So this gave a quick explanation, or a first explanation, of the different defects that might be involved to generate the immune deficiencies that we were seeing inherited in children. So the X-linked agammaglobulinemia in this light could be on the basis of an early block in development of the B lineage. We knew of examples like the Wiskott-Aldrich, where the block in development of the cells that were responsible for cellular immunity could be based on a thymic defect, and very soon a demonstration by a pediatrician named George of children who were born with maldevelopment of these pharyngeal pouch epithelial that provided the epithelial thymus, and also the parathyroid gland, so that they had calcium disturbance and tetany at birth, and had absolutely no thymus, but still had plenty of plasma cells and antibodies that were producing, plasma cells that produced antibodies. There were other so-called Swiss types of inherited agammaglobulinemia that resulted in deficiency of both T and B cells, and then even earlier defects. And this gave an early roadmap for replacement therapy, with providing the progenitor cells from bone marrow, or grafting in a thymus in case of the DeGeorge syndrome with this particular block at number 4. It also, there was information that showed that lymphomas or leukemias, like the ones that Dr. Miller will tell you about that arise in the thymus in mice, can be of either lineage. One of the clues for that is a virus-induced lymphoma in birds. So if you give the leukemic virus at a day of hatching, as had been shown, about 6 or 8 months later, they develop lymphomas. And a colleague of mine, Ray Peterson, had shown that if you remove the bursa, you could prevent the development of those lymphomas, whereas the thymus had no effect, very unlike the mouse model. In looking at the time related after the giving the virus, we could see that what happens is that there was a multi-stage process in development of these malignancies. even as early as 1 month, there were already follicles that were overgrown, but very few of the thousands of follicles that were there. And it took about 6 months before these B lymphomas were generated. Since then, it's been shown that where the virus is inserted, near oncogenes and what mutations occur, oncogenes like MYC and MYB, depends on one of the different stages in this process. So these sort of clinical manifestations were quickly apparent, but there were fundamental questions that were asked by this two-lineage model. One is, if the T cells can't make antibodies, what do they use to recognize antigens? Another is, if there is B cells can't make antibodies normally without T cells, what is the basis of this cooperative interaction? And a question that we addressed for some time, since we mammals don't have a bursa aphabresis, where are B cells being generated in us and in other mammals? And we spent the next decade looking for this bursa equivalent. Before finally, with John Owen and Martin Raff, John had devised a way. They thought that B cells might be generated in hematopoietic tissues. And to test that, John Owen had developed a way to culture liver, fetal liver, where hematopoiesis occurs in late-stage mouse embryonic development, to grow them under conditions that the cells could survive. And so we cultured the cells, took them out a week later. We cultured them before they had B cells. A week later, we could see that they had generated B cells with antibodies on their surface, indicating that they could develop in these tissues. Later, in contemporaneous studies, Osman and Nossel, and also other scientists in Switzerland, Vasily and his colleagues, could show that cells in adult bone marrow that didn't have surface antibodies could give rise to ones that did express surface. So this suggested, and we could show as well that we could culture bone marrow from mice before hematopoiesis started, and those cultures would generate B cells as well in bone marrow. So it became obvious that these were generated in hematopoietic tissues. Now, in some of the animals, we had shown in earlier studies that earliest B cells that express in chickens the surface antibodies, they're vulnerable to ligation resulted in cell death, in studies done by Paul Kincaid in our laboratory. And we could show in these cultures of this fetal liver from mice the same thing. We could get rid of the B cells by treating with antibodies to IgM on their surface. But when John Owen thought there were still lymphocyte-looking cells, and when we fixed the cells and stained them for intracellular elements, they still had expression of IgM components. Now, my colleagues later realized that they were only making the heavy chains of IgM, not the entire IgM molecules. And by using radiolabels and doing studies as a function of time to follow development of cells that didn't express anything of heavy or light chains in there, became cells that expressed the heavy chain, and those give rise to the B cells, and we could track the time that it took to follow this pathway. Later, Susumu Tonegawa, of course, cloned the immunoglobulin genes, and the rearrangements from D to J and then V to G, J for heavy chain expression and V to J on the light chain genes accounted for this asynchronous expression of heavy and light chains. Sakaguchi and Karasuyama, who discovered these surrogate light chains on pre-B cells, allowed tracing the development. And we could use these markers to show similar pathways of B cell development in rabbits and humans, for example, as well. Now, I won't go into that and the isotype switching that occurred, or the T cells that Dr. Miller will tell us about shortly. But I'll go quickly for my last few slides to address another question that we've been trying to investigate in recent years. And it started with Jan Klein, a famous genetic immunologist who did important work in major histocompatibility gene evolution. We wanted to know what the roots of our adaptive immune system was, and which came first, B cells or T cells. Now, all other investigators have shown that every jawed vertebrate, all the way from cartilaginous fish like sharks, have T cells that are generated in the thymus, B cells that are generated in hematopoietic tissues, the same genes that are involved in the generation of the diversity of the different immunoglobulin gene segments. But we couldn't look at earlier jawed vertebrates because they all died out. We only know about them through fossil remains. But there are two jawless vertebrates that are still surviving, lampreys and hagfish. And so Jan Klein and I went back because by this time we could get sequencing. We could isolate cells that looked like lymphocytes by flow cytometry and express cDNA to see what transcripts and get sequences by what was then high-throughput sequencing. And we couldn't find any of these cardinal elements that we use for our generating our T and B cell receptors or MHC class genes. So we went back because we knew that others had shown that if you immunize lampreys or hagfish with red blood cells or bacteria, they do make agglutinins, and they seem to be specific, and they can reject grafts, albeit slowly. So we thought maybe if we catch them in the act of responding, so we stimulated them with a cocktail of live bacteria, plant mitogens, and then looked at the expression patterns, gene expression patterns for the lymphoblast, and we started seeing lots of leucine-rich repeat molecules that looked a little like this cartoon. With part of it was leucine-rich repeats on this end, and that was highly variable. And whereas the other end, the stalk region over here on the right, was invariant. And so we cloned and sequenced lots of these from different animals. In fact, we sequenced Zeb Panzer and I hundreds of these, and virtually all of them were different from each other, as shown by this, in sequence, as shown by this phylogram. The other thing that we learned, and others saw as well, is that if we did a single cell analysis, they only express one gene for one of these variable lymphocyte receptors. And this is a schematic model of what we've learned about that. These don't have lots of genes encoding these variable lymphocyte receptors. In fact, the genomic sequence is incomplete. It only encodes for the ends of this leucine-rich repeat part and not the middle, and this invariant stalk region. But flanking that gene, we could find lots of coding sequences or cassettes of leucine-rich repeats that, if copied in here, and that happens on the basis of homology or similarity between a donor and a sequence in the germline gene, and it happens in bits and pieces over and over and over, starting on one end or the other, and goes through until you get a complete gene. And when that happens, the cell expresses it on the surface, can be stimulated, and the repertoire we estimate is greater than 10 to the 15th, which is as great as our repertoire. And it turned out since Dr. Kasahara found a third class, and we found a second class of these. There are 3 of these germline incomplete variable lymphocyte receptor genes. We call them A, B, and C. B is what we've been talking about, that's expressed by these B lineage type cells. The other 2 are expressed by cells that in a thymus equivalent at the tips of the gill folds, that like the thymus, they assemble the VLR-A and VLR-C, and they look much like our alpha, beta, and gamma delta T cells in their genomic program. So that tells us or suggests very strongly that this pattern of having 2 prototypic types of T cell lineages and 1 B lineage in type, that genetic program was derived, was already present in a common ancestor of jawless and jawed vertebrates somewhere around 500 million years ago. So T and B cells have been constant companions ever since, and probably for reasons that allowed survival. And one last point, the antibodies that are made in multiple multimeric forms by lampreys and hagfish have these leucine-rich repeat binding sites that are absolutely specific. In each case, they are more specific than a comparable high-affinity antibody made by a mouse or a human, perhaps because of the structural rigidity, and also they can make antibodies that are rarely seen among our antibodies or mouse antibodies, perhaps because they don't have the tolerance constraints of seeing those antigens that are present in us. And we're hoping that those will be biomedically useful for diagnostic and possibly even therapeutic purposes in the future. These are my research colleagues, some of whom I mentioned, or at least referred to by along the way, and many people contributed to these studies. And thank you very much for your listening to this long, fishy tale.
Hello. I'm very grateful to the Japan Science Foundation for including me in the JapanPrize, and I'm very happy to share it with my longtime friend, Max Cooper. Thank you very much. So what I'm going to do, I'm going to go through the events which led to the discovery of thymus function and to the identification of the 2 major types of lymphocytes called T and B cells. As you probably all know, the thymus is situated in the chest, where it shows here, above the lungs. And for a long time, its function was totally unknown. The function of lymphocytes, which are a type of white blood cells which circulate in the blood, was known at the time that I started work. When these lymphocytes were confronted with antigen, which means something foreign, by the way, I'm going to talk non-scientifically as much as I can. I'm going to leave out as much as I can the words which are technical. So when lymphocytes meet antigen, which means a foreign body, they become changed and they form antibody, and they also can reject foreign skin. That's called cell-mediated immunity. And it was discovered in the late 1950s that if you took blood and took a lymphocyte, a cell, a white blood cell from the blood, there it is, and you label it and you put it back in the blood, it circulated right round. However, if you took the same type of cell, because the thymus is made up of lymphocytes, if you took the same type of cell from the thymus, injected it into the blood, it just ended up in the spleen and died. And this, plus the fact that thymectomy in the adult did not show any changes, suggested to most people that the thymus has nothing to do with the immune system. No immune function whatsoever. And the conclusion was made by a Nobel laureate that we shall come to regard the presence of lymphocytes in the thymus as an evolutionary accident of no very great significance. So about this time, I was going to the Institute of Cancer Research in London. I wanted to do a PhD, and I did it here at the Institute of Cancer Research. I couldn't get a space in the main institute in London, so I went to a satellite institute, as you can see here in the country. It was very beautiful, but my lab was in the shack, and my mice were bred in horse stables. Now that's very important because horse stables are not very clean. And part of the reason why I discovered the function of the thymus was because my mice were not clean. Remember that. So first of all, I want to tell you a bit about mice. I decided to work on mouse leukemia. There are different strains of mice. For example, the brown strain is an inbred strain, so any brown mice will not reject brown skin. The black mice is another inbred strain, so any black mice will not reject black skin. But if you put skin across the two, you will reject it. So that's the law of genetics. And what about leukemia? Leukemia starts in certain strains of mice. This one's called the white mouse. It starts in the thymus, and it goes to it after about 1 or 2 months. It sort of migrates to the rest of the body. But at the time that I came to London, Gross had shown that you can extract from the thymus what he called was a virus. It is a cell-free extract, which when given to mice of a low leukemic strain, mice which don't normally get leukemia, it would give them leukemia. However, there was one caveat. It had to be given to mice of low leukemic strain very soon after birth. That's why I've got a newborn mouse here. So I repeated Gross's work, and in fact, it's exactly evident. If you give it at 1 day of age, you get leukemia. If you give it at 2 months of age, you don't get leukemia, only in 17%. And if you give it later, you get no leukemia at all. So, and leukemia begins in the thymus. So there's two questions. One, why should you give it at birth, the virus? And two, why should the thymus be involved? So I decided to study this problem. So here is, because it begins in the thymus, the obvious thing was to do an adult thymectomy, take out the thymus. So these are sham thymectomized mice, which means that they just open the chest, don't take out the thymus, and then close the chest again. And after, at 6 weeks of age, you can see the adult thymectomy prevented leukemia completely. Well, that makes sense because leukemia begins in the thymus and then migrates. But I did something else. I put the thymus back, as you can see here, immediately after thymectomy, and you get leukemia back. However, if you wait, instead of putting it back immediately, you put it back 6 months later. You wonder whether you still get leukemia. Well, you do, as you see here. So clearly, the virus that you injected at birth must have multiplied and must have been hiding somewhere, waiting for the target, which is the thymus. So I thought, how to resolve this? Why should the thymus be, why should the newborn mouse be involved, and why should the thymus be involved? So the questions I ask are shown in this. This is a hypothesis, this is expected results. If the virus must multiply in 1-day-old thymus, well, take out the thymus before 1 day, at birth. Take it out at birth, and then give the virus. The virus can't multiply anywhere, it's just flushed out. So when you come along much later, say 6 months later, and put the thymus back as a graft, well, you shouldn't get anything. If, however, the virus can multiply anywhere else, then you don't need a thymus at all. You would put the thymus back any time later, you should get leukemia. So that's the reason why I did thymectomy in a newborn mouse. And I didn't expect the results we are getting now. Look at this. So this is the newborn thymus. As you can see here, my thumb and index finger gives you an idea of the size. So I took out the thymus from newborn mice, and nothing happened. They grew perfectly well for the first 4 weeks of life. You can see them growing perfectly well. But then something happened which never happened after adult thymectomy. And here it is. Mice which had been thymectomized at birth started to get sick about 12 weeks of age. And they wasted away. So I had a look at their organs to see what was happening. And this is what I saw. Lesions in the liver suggesting an infection by hepatitis virus, which normally does not happen in normal mice. So something was wrong with these mice. And then, of course, I looked at the lymphoid tissues, and the lymphoid tissues were deficient in some areas of those lymphocytes which Gowans had shown were recirculating. So it was clear that these mice had something wrong with their lymphoid system and therefore their immune system. So what I did is simply to test for the capacity to reject skin grafts, because skin grafts, foreign skin grafts, are rejected by an immune response. And obviously here you can see a red skin graft is not rejected, another skin graft from a different strain of mice is not rejected. So obviously these mice had difficulty in making an immune response, so it's obvious that these mice were deficient in their immune response. So the thymus must have had something to do with producing lymphocytes which recirculate. So then I did thymus grafts from a foreign strain to see if the thymectomized mice would become tolerant of the foreign strain. Normally they reject foreign strain skin, but if they have a thymus from foreign strain, the question was, do they accept a thymus from the foreign strain? They do, or do they accept it from another strain? They don't. So clearly the mice were tolerant of tissues taken from the thymus donor, suggesting that the thymus not only produces cells which are important in immunity, but makes sure that cells which can react against yourself are deleted. Also, they fail to produce antibody, as you can see here. So not just rejecting graft, but they fail to produce antibody. And at that time, we had no markers whatsoever. The only markers we had were those that I mentioned, which are dictated by histocompatibility differences. The other marker we had was a T6 strain, which has a tiny chromosome marker as shown there. So that was very useful in the days where we had no other markers. And I could show then that the thymus can produce cells, which, here's a marker here, which when they grow up and after an active immune response can be found in the lymphoid tissue. So the thymus is producing lymphocytes which go out into the periphery and eventually recirculate. The other thing that I asked the question, I mean, adult thymectomy had no function whatsoever. It didn't worry mice. After adult thymectomy, they were perfectly all right. But that's probably because they had lymphocytes already in the circulation. So the idea was to get rid of those lymphocytes. So I did that in 1962, got rid of all the lymphocytes by irradiation, as shown here, and had to protect the mice with bone marrow, because bone marrow is a source of stem cells, which gives rise eventually to lymphocytes. But mice without a thymus could not reject foreign skin grafts. So adult thymectomy is doing nothing unless you irradiate them. Now this shows that the adult thymus can even repopulate the lymphoid system, provided the lymphoid system has been destroyed by irradiation or something else. And then the other thing that I showed was that mice that had no thymus since birth were much more prone to develop cancers than mice which are normal. As you can see, mice which are normal do develop some cancers, but not as much as mice which have been thymectomized at birth. So just to summarize, this is what happens for mice thymectomy birth, as I mentioned. They are tolerant of thymus donor strain. The adult mice don't show any immune defects unless you previously irradiate them, and some thymus lymphocytes do emigrate. Now, the immunological community did not like it that because they thought the thymus had no function whatsoever. So this is what they say. Mouse trends must have been unusual. If it happens in mice, it can't happen in humans. Mice used in the converted host tables, and here it is, must have been exposed to so many intercurrent infections that the additional trauma of neonatal thymectomy or irradiation precipitated immunodeficiency. So it's because of this criticism that I went to NIH, the only place in the world in 1963 where you had germ-free technology, germ-free mice. So I repeated all the work in germ-free mice. Here you are, I'm skin grafting in neonatal thymectomy mouse, and this is what happened. The mice are perfectly healthy, they don't become sick, they can't get infected because they are germ-free, but they cannot reject foreign skin, as you can see here. So obviously, the neonatal thymectomy does interfere with the capacity of the lymphoid system to make lymphocytes. So that was accepted after this experiment. Now, as Max said, the chicken has its thymus and has a bursa. So what's happening here? Burnett and his colleagues, as Max alluded to, Schoenberg and Warner, did the following experiment. They used testosterone to get rid of the bursa or bursa function, and these mice could not produce antibody. And in some of the chickens, the atrophy extended to the thymus, and in these chickens who were rather sick, they failed to reject skin. So Burnett asked the question very quickly, what is the functional equivalent of the bursa in a mammal? Because mammals do not have a bursa, of course. And then he suggested that in mammals it is highly probable that the thymus also carries out the function of the bursa, which is to feed into the body the cells whose descendants will produce antibody. So I thought that was good. I thought thymus must produce cells which produce antibody as well as cells which are responsible for removing virus infection or any type of infection or getting rid of skin grafts. So what I did with my first PhD student, Graham Mitchell, was simply to look at the recirculating lymphocyte pool. And you do that by cannulating the thoracic duct, which is the main lymphatic duct, which collects all the lymphocytes. So you cannulate the main thoracic duct, and you collect all the lymphocytes. And you can see that this graph becomes flat, which means that you've collected all the lymphocytes out of the mouse that you can. If you compare that to neonatal thymectomy mice, you find there's a tremendous difference, as you expect. So the pool of circulating lymphocytes after neonatal thymectomy is not being built up. So the top one must be thymus-derived, and the bottom one may be derived from some other source. Anyway, we did prove that the lymphocytes collected at the top were indeed thymus-derived. So now we had a way of asking the question, what produces antibody? We wanted to show that the thymus-derived cells, which are here, thymus-derived lymphocytes, are the ones which produce antibody. So we used two strains of mice, A and C, and you breed those two, and you get a first-generation hybrid A cross C. Now what happens is A cross C, thoracic duct lymphocytes, which you take out by cannulating the thoracic duct, are injected into adult, thymectomized, irradiated, and bone marrow protected A cross CF1 mice. These mice here are totally immunocompetent because they've been thymectomized, irradiated, but they are going to die because you need a source of stem cells. These mice will not survive unless they are given a source of stem cells. So we gave them bone marrow from one of the parental strains intravenously. Now, this was done in 1967 and 1968. And then we challenged the mice with sheep white blood cells, and we got antibody-forming cell, which we could detect on agar plates, because the agar plates were overlaid with sheep red blood cells, which are red, and they make if you make an antibody, you see a plaque, like you see those white plaques. Now we ask the question, where do the plaques come from? If they come from the thoracic duct lymphocyte, which is thymus-derived, you should be able to get rid of them with an anti-C serum, with an anti-8 serum. If they come from bone marrow, they should be able to get rid of with an anti-C serum. So this is what happens with anti-C serum, as you expect, because C is present in both A cross C and B, and C, right? Now, the crucial one is anti-C serum, because the C antigen is present only in the thymus-derived cells. So if the antibody comes from the thymus-derived cells, then it should be eliminated. You should get exactly the same as here. And what we got was that. So clearly, it's very clear, the results were perfect. Repeated every time. The antibody-forming cells do not come from the thymus, do not come from thymus-derived lymphocytes. They come from bone marrow. And this was shown in 1968. So thymus-derived lymphocytes do not form antibody. Bone marrow-derived lymphocytes do form antibody, but because I had shown that thymectomized mice have a difficulty in making antibody to a large number of antigens, then clearly there must be a crosstalk between T cells, thymus-derived cells, and B cells, bone marrow-derived cells, to produce antibody to many antigens. So what we suggested is that the B cells see some part of the antigen and the T cells see another part. If it doesn't fit, it doesn't come. If it fits, then it would give some kind of signal to the B cell to make antibody. So this was our position in 1968. However, since 1968, a lot of people, millions of people, I could say, have been working in immunology, and we know much more about this than we used to know in 1968. We know that there is a cell, a dendritic cell, which is necessary to gobble up fragments and to present the fragments to T cells. So here I have tumor cell fragments, but it can happen with any type of fragment, any type of antigen. So it gobbles them up, it digests them, and it presents them to the outside in this molecule here called MHC. And the T cell that comes along that can see the antigen on the MHC, which is presented to it, can be activated provided it puts out another molecule, which is shown here, CD28, which binds to the dendritic cell. So as a result of those two binding molecules, then the T cell can be activated. However, the T cells cannot remain activated forever, because if it did, it could cause collateral damage. It could cause too much inflammation. It could cause autoimmunity. So nature has made sure that after being activated, the T cell has to be switched off. And it is switched off this way, by this molecule and by another one. Doesn't matter their names. They're just molecules which tend to switch off an immune response so that there's no collateral damage. Now, what has happened is that cancers have utilized this mechanism to make sure that the T cell is always switched off. Here's a cancer cell. It puts out a molecule which makes sure that this molecule is always on, so that the T cell is always switched off, so it cannot kill the cancer cell. That's what's happened. So for many years, we did not know that. We did not know that. And this is the reason why we couldn't cure cancer with lymphocytes. But now people have realized that this molecule has to be, we have to stop these checkpoints, and people like James Allison in Texas, or now somewhere else, have made antibodies which bind to, oh, sorry, there's another cell which I didn't bring in, another cell discovered by Sakaguchi, who's here, which is very important in switching off activated T cells, which are killer cells. So, okay, the cancer recruits this other cell, which is called Treg cell. And so Jim Allison and others have made antibody against these checkpoint molecules, TLA4 and PD1. And because now the cancer cell can no longer recruit the Treg cell and can no longer see the T cell. The T cell now can migrate and kill the cancer cell. There's no way the cancer cell can escape. But this doesn't happen to all cancers, unfortunately. So we must find out why some cancers are not killed by T cells. And we can look at the molecules, more molecules. I mean, I could go on and on and on and on and tell you about other molecules that may be involved. So we have to do much more work to find out how to make sure that T cells can kill every type of cancer, or as many types as we possibly can. Okay, now I'll just show you an example. And here's a woman on the left who's got a melanoma, which is very common in Australia. And this melanoma has not been touched by surgery, not by radiation, not by chemotherapy, just by injecting the antibody I told you about, which stops the checkpoint. And in 12 days, it's all gone. Now, the woman also had cancer in her liver and in her lungs. And the T cells, which had been activated by this mechanism, have gone round the body, and within 5 weeks, she was completely clear. So I think the message of this talk is simply to say that basic research on T cells has spawned immunotherapy, which now can claim a seat in the therapeutic pantheon of oncology, next to and probably about to supersede surgery, radiotherapy, and chemotherapy. Thank you very much.